Hello, I'm Richard Anderson. I am the PI on NIH BRAIN Initiative and NIH-funded research on advancing human neuroscience through the development of neuroprosthetics. Today, members of our team will be discussing how multidisciplinary research can advance clinical and basic research of the human nervous system. The title of our panel is Thought to Action, Developing Brain-Machine Interfaces to Assist Individuals with Paralysis. So a brain-machine interface essentially consists of three components. There's a recording electrode array, a decoder, and an assistive device. So the recordings are made of neural signals. Those signals are decoded to determine the intent of the individual, and then those are formed into a control signal to control assistive devices such as robotic limbs or computers. The implant that we use is a tiny array of 100 electrodes. It records 100 to 150 neurons, and then the decoder uses the information from those neurons to determine the intent of the individual. Now, the types of paralysis that can benefit from brain-machine interfaces are patients with spinal cord injuries, stroke, multiple sclerosis, amyotrophic lateral sclerosis, and Duchenne's muscular dystrophy. Participants in our study have tetraplegia from high-level spinal cord injury. So the incidence of this is quite high. Around 250 to 350,000 people in the U.S. currently have spinal cord injury. About half are at the high cervical level. Our participants are all at the high level injury at cervical levels C3 through 6, and they are tetraplegic, meaning that all four limbs are paralyzed, and they've also lost feeling in those four limbs. There are very few assistive technologies for them, and they have very high dependence on caregivers. This research is by definition multidisciplinary, and it first of all includes our participants. They work very hard, they're very dedicated. Without them, this research wouldn't be possible. And they do this selflessly, not to benefit themselves, but for those that will follow as the technology develops. We also have at Caltech a Chen Brain-Machine Interface Center that includes our laboratory, and we collaborate as well with the Shapiro Laboratory and Imami Laboratories here at Caltech on technical advances. Also for technology, we work with the Tanter Lab in Paris. We, of course, must work with the clinician. We work with neurosurgeons at Keck USC Hospital and at the UCLA Medical Center. It's very important to have restoration hospitals involved. For us, it's Rancho Los Amigos and Casa Colina, both rehab centers in the Los Angeles area. And then finally, we also collaborate with other universities and hospitals on scientific projects. Today, we'll have four key investigators from our group speaking. There's myself, and I'm trained in neurophysiology, and Spencer Kalis, who's at Caltech and USC, trained in engineering, Tyson F. Flallow, trained in neural computation here at Caltech, and our clinical colleague, Charles Liu, who's a neurosurgeon at USC. Initially, we began our work in non-human primates, understanding visual motor integration in the cortex, and we found that vision cortical areas project into the posterior parietal cortex, where intentions to move are formed, and that projects then to motor cortex. And we found an area that was specialized for reach, another for saccades, Sakata and colleagues found an area that is specialized for grasp. And from this, we proposed that there was a map of intentions within the monkey cortex, and that possibly something similar would be found in human. And we could record the intention of humans in a prosthetic application and use it to help paralyzed people. Now, this next slide shows the basic problem of breaking the loop between vision and motor output with spinal cord lesions. So you can see an object and process it through visual cortex, posterior parietal cortex for intent, the motor cortex to send a command signal down to the spinal cord for making a movement. However, if the spinal cord is severed, then the muscles can't be activated. So groups have recorded from motor cortex to get the execution signal for prosthetic applications. This makes a lot of sense because the motor cortex is the final motor output from the cortex to the spinal cord. Since we've had, and by the way, from motor cortex, you can get the trajectory of the limb movement, which is the one limb on the contralateral side. From our experiments in posterior parietal cortex looking at intention in animals, we found that not only could we get trajectories, but also the goals of movement, and this can be very fast. We also found that the cells code for movements of both limbs, and that there were neurons specialized for different grasp shapes. So a single neuron can convey an entire grasp. So 6 years ago, we implanted our first subject, Eric Sorto, and we, in our studies, we asked the subjects what they would like to do as a goal within the study. And in Eric's case, he wanted to be able to drink a beer at his own rate. In other words, not have someone do it for him. And so in the video that you'll be seeing, this is the first time he was able to drink a beer at his own rate. In this case, he was controlling the timing of the sequence of the robotic movement with his brain signals. Since then, he has been able to use his brain signals to identify the goals of a movement and the action that he wants to perform on those objects. So if you could please now run the video.
All right.
Hey, you finished that thing off? That's good. And that's how science is made. All right.
Yay!
There you go.
Did it.
So we found that the variables that we thought we would find from the monkey research was there in humans as well. So we could decode goals and trajectories and also grasp shapes. However, we found many, many more variables as well. We could decode the movement of individual fingers. We could essentially decode the intended movement of all of the body, any part of the body. And we also found we could decode action observation and action semantics. So a question became, how is this possible? How can recording from so few neurons in such a small patch of cortex enable us to decode so much? And the answer we found in experiments that were designed to see how information was encoded in the posterior parietal cortex. So this is one example for action semantics. In this case, we showed the subject videos from 3 different points of view of subjects performing actions on objects, and also showed them in a 4th format, a verb for the action, in this case, grasp. And we did this for 5 different kinds of actions: to operate on objects, to drag them, drop them, grasp, push, or rotate. And this one neuron that you see on the left was active for rotating an object from any perspective, and also by silently reading the word rotate. So this would be a neuron invariant to the particular action type. And here's a cell that's similar, but not with the language component. Here's one that has rather a mix, and additional one with a mix of various actions. And here's one that is active only when reading the word grasp. So what this shows is that for, in this case, this particular class of experiments, action semantics, there's a mixed selectivity in the population of neurons for the action type. This is another experiment examining this mixing. Here we're looking at mixed selectivity for the left and right hand for 2 different actions and speech. So it's attempted movement of the hand, imagine movement of the hand, or to speak left or right. And here is a neuron that is active for attempt and imagine the right hand moving. This next one is for attempt just of the left hand. This 3rd neuron is active only for imagining left or right hand movements. And this final neuron is active when the subject says left but not right, and otherwise is not active. And this is a similar experiment, except here we've replaced speech with shoulder movements. And again, we're looking at strategy of imagine versus attempt, and side of the body, left and right. And effectors are now the shoulder and the hand. And one possible way of mixing is shown in the upper part of the panel, in which everything is randomly mixed. And we found that this is not the case. Actually, there's a structure to the mixing. So there are different subspaces for the effectors, one for shoulder and one for hand. And within those subspaces are overlapping strategy and side of the body representations. So one advantage of the mixed representations for the side and strategy is that there's an overlap in learning. So it's been shown that you can learn something with the right hand that more easily transfers to the left hand, or you can use imagery to improve motor performance. An advantage of the segregation and representation of the shoulder and the hand is that shoulder movements are very different from hand movements. And this, thus, there's little advantage and potential interference from having them overlapping. Now, for the semantics encoding of action type, we likewise found this structure. So the segregation was along action type. For instance, the grasp verb and grasp videos are partially segregated from those from the other actions, such as rotate. So this is basically the answer to how so many things can be represented in such a small number of neurons. It's this partially mixed selectivity that enables all these variables to be encoded across the population of cells and can be extracted with the decoder. Current extensions of our research are intracortical microstimulation to restore the somatosensation that's lost. Spencer Kalis will talk about this. And the question here is, can artificial sensation be natural and improve dexterous control of a robot hand? The talk by Tyson Aflalo, will it be a direct comparison between the posterior parietal cortex and motor cortex? And one question that he'll be addressing is whether it's important to pick the right cortical area based on the intended applications, or will plasticity allow any cortical area to suffice? We'll also be looking in the future on whether the posterior parietal cortex is indeed higher dimensional than motor cortex. Other studies that we're doing, but we won't talk about in this panel, are using functional ultrasound. This is with Mikhail Shapiro and Mikhail Tanter labs. And the aim of those studies is to develop minimally invasive brain-machine interfaces for use in humans. Also, we are looking at machine learning decoding with Azita Amami's group here at Caltech. They are seeing if they can design decoders on microchips that can be implanted in the brain and learn to decode neural signals. Thank you very much.
Hi, my name is Spencer Kellis. I'm a member of the professional staff at Caltech and a research assistant professor of neurosurgery at the Keck School of Medicine of USC. And one of the benefits of working in both institutions is the opportunity to work closely with a multidisciplinary team of physicians, scientists, and engineers. In this talk, I'll be discussing how we're able to leverage these opportunities to work and to understand generating somatosensory percepts with a broader set of patients and a broader set of electrode types. At Caltech, I'm involved in a clinical trial of brain-machine interfaces. In that trial, we have one tetraplegic participant, FG, who has microelectrode arrays implanted in ventral premotor cortex, or PMV, supramarginal gyrus, or SMG, and the somatosensory cortex, as highlighted in red in this image on the right. These microelectrode arrays are manufactured by BlackRock Microsystems. They're very small devices. As you can see in the lower right, they're about 4 millimeters on a side, which means that 100 electrodes fit into the fit into an area smaller than the size of a penny. And that means when we're stimulating through these electrodes, we're targeting a very small area of cortex, and we use very small amplitudes of electric current for the stimulation. At USC, we work with epilepsy patients who are undergoing surgical treatment for their epileptic condition. Now, these patients have a different kind of electrode implanted. These are called electrocorticography grids, or ECOG for short. And as you can see in the picture on the right, these are essentially just a grid of contacts that rest on the surface of the cortex. So there are two different kinds of ECOG grids. The larger one is the standard clinical grid. These are 5 millimeter contacts with 1 centimeter spacing. And then there's a smaller, denser version, which we call mini ECOG. And these have 2 millimeter contacts with 3 millimeter spacing. And so the larger one at 7 centimeters across and the denser one at 2 centimeters across are still both much, much larger than the 4 millimeter across microelectrode array from the previous slide. And so when we stimulate through these contacts, we use larger amplitudes of electrical stimulation, and we target a larger area of cortex. So we are working on a bidirectional brain-machine interface. And what that means is that we are trying to record neural signals of intent from parietal cortex or motor cortex. And we interpret those signals or decode them to control an assistive device, such as a robotic hand. And then that robotic hand might have sensors embedded, for example, in the fingertips, by which it can acquire sensor data when it interacts with objects in the environment. And our goal then is to be able to encode the sensor data and deliver it back to the brain by electrically stimulating the cortex. So this is the area or the aspect of bidirectional brain-machine interfaces that I'll be discussing today, this problem of how to encode somatosensory information and deliver it back to the brain. To do that, I'll be first showing a clip of an interview with a man named Ian Waterman, who at the age of 19 lost all sensation, but was not paralyzed. And so he's in the unique condition of being able to move, but not being able to feel. And in this video clip, he discusses how difficult it is to do simple everyday tasks, such as holding a cup, without being able to feel. Play video.
And sound, whenever I'm working with my hands or doing something with my hands, I'm not just looking and thinking about hands, I'm also attending to sound. Yeah. I mean, I can't get anything from my pocket because I can't feel it terribly well. You know, I can't do something I can't see. Often if someone passes me a cup, I would grab it awkwardly because I don't have the feedback of something coming from the fingertips saying you've actually picked up the cup incorrectly or taken it incorrectly.
So as you heard in that video, he described how difficult it was to do these simple tasks such as holding a cup without having any sensation at all. This really captures what we're trying to do with our clinical trials. We're trying to be able to restore the ability to sense as well as to control these assistive devices. So next I'll play a short video from our tetraplegic research participant FG while he experiences some electrical stimulation to the somatosensory cortex. So you'll notice in this video that FG describes the sensations he feels in his own words. And he'll also report the intensity of the percept that he felt on a scale of 1 to 10, and the duration of the percept he felt, short, medium, or long. And as you watch this video, you'll see in the main portion of the screen, you're watching essentially what he saw on the computer screen. So there will be cues to the different phases of the task, the intertrial interval, or ITI, the stimulation with the purple dot, then a delay, and finally the response. And on the right side of the video, you'll see representations of the two different arrays implanted in somatosensory cortex to indicate which electrode is being stimulated during the stimulation phase. Play video.
Yeah. W13, short, move forward, about a 1. Yeah. It's 7 and it's 8. Medium grab, about a 7. Yeah. E13, it was a short tap, about a 2.
So you can see in that video that he was able to identify very specific locations on his body where he felt these percepts. And so this was one of the very first questions we wanted to investigate. Where did he feel these percepts versus which electrodes were we stimulating on? And so here in this figure in panel A on the left, you can see sort of the receptive fields of where he felt these percepts. And they tended to be focused primarily in the upper arm in green and in the forearm in pink, and then a little bit in the hand in blue. And then if you look at the panel B, which shows you which electrodes corresponded to which area of the body, you'll see that the medial array tended to cluster responses to the upper arm, whereas the lateral array tended to cluster responses to the forearm, which generally follows the homunculus as you might expect. So then we also wanted to look at the same question with the ECOG epilepsy patients. And so here you see again this dense mini ECOG grid. And then there in the middle, there's a reconstructed image of a brain. And so on that brain, you can see highlighted in blue is the motor cortex, highlighted in green is the somatosensory cortex. And then the magenta dots are the mini ECOG grid contacts. And they're resting right over the hand area of somatosensory cortex. And so with one representative subject, which went through this experiment, you can see that this subject was able to identify or experience percepts in each of the digits on the hand. And then if you look at which electrodes tended to generate those percepts, there is a clear relationship between a specific electrode and a specific digit. And then across all 9 subjects that participated in this experiment, we found similar observations where each of the 9 patients was able to identify receptive fields in the hand. And so clearly in both modalities, with microelectrode arrays and with ECOG grids, it's possible to generate a percept in a very specific location. So it's also interesting to look at what perceptive qualities people feel when they're stimulated in somatosensory cortex. And so here I've listed the most common descriptive terms that people use from both modalities. So for ECOG, stimulation through the ECOG electrodes, people reported sensations such as a tap, a tingle, tremble, tickle, buzz, electricity, itch, pulse, shock, and even movement sometimes. Whereas for the microelectrode arrays, there's a tap, vibration, squeeze, goosebumps, press, blow, pinch, and then upward movement, right movement, and forward movement. And so a couple of things to note here. One is that with the microelectrode array, the terms that were used, that FG used to describe these percepts tended to be more naturalistic than with the ECOG, stimulation. And then secondly, it's interesting to note that FG reported several instances of movement kinds of percepts, which are a proprioceptive-like quality. Now, when we stimulate using a biphasic charge balance pulse train. And an example of what that looks like is shown here in black. This is the square waveform. There are several aspects of this waveform that we can control when we deliver stimulation. For example, the amplitude, the pulse width, or PW for short, the frequency at which these pulses appear, and then the total duration of the pulse train, as well as the electrode that we stimulate on. And this becomes interesting when you start to look at what changes in the waveforms correspond to what kinds of changes in the perceptual qualities that patients experience. So I mentioned earlier that our subject FG reported some movement or proprioceptive-like qualities in some of the stimulation trials. And here I'm showing how those map onto the different frequencies of stimulation and the different amplitudes of stimulation that we tested with him. As it turns out, the proprioceptive responses, which are shown in blue as a percentage of the total responses at each amplitude and frequency, tended to occur at higher amplitudes, but across all frequencies, relative to the cutaneous percepts, which occurred at across all amplitudes and frequencies. And then if you look back onto what electrodes generated these proprioceptive versus cutaneous percepts, there's not really necessarily any clustering to one array or the other. And so this tended to be across both arrays. With ECOG, on the other hand, we found that higher stimulation parameters in general led to higher reported intensities in terms of how the patients described what they felt. So this is an example from two patients in the ECOG study, subject 6 and subject 3. And we're looking specifically at amplitude now. So the four different columns indicate stimulation with amplitudes of 0.5 milliamps, 3 milliamps, 5 milliamps, and then finally 10 milliamps. And as the color gets darker, that indicates a more intense reporting. And then you can see for subject 3 that the location of the reported percept tended to change as the amplitude increased. So here are the results for frequency for the same subjects in the middle, and then for pulse width for the same subjects in the middle. And the overall takeaway here is to see that as all three of these parameters increased in value, the patients tended to report higher intensities in whatever the perceptual quality that they described. So now an important aspect of sort of closing the loop here is to identify whether it's possible for people to use this information to complete a behavioral task. So we did two of these with the ECOG epilepsy patients. And the first, which was a simple left-right discrimination task, patients simply held their hands out over a piece of paper with two dots on it. They were not colored differently in the experiment, except that one of them, when the patient held his hand over that particular dot, corresponded to receiving stimulation and the other did not. And the patient's instruction was to identify which dot corresponded to stimulation. 8 out of the 9 subjects participated in this task, and there was on average 100% accuracy in identifying the correct dot on the paper. In a slightly more complicated task, patients saw a paper with a grid of 9 dots in a 3x3 array and had to identify the orientation of a line consisting of 3 dots through that grid. And so in this image, you can see one example trajectory as one of the patients held his hand over the grid and started searching for this line that corresponded to stimulation. And so again, out of 8 of the 9 subjects that participated in this task, there was an average of 98.3% accuracy. And so clearly, these patients were very much able to use this information to complete these relatively straightforward behavioral tasks. So over the course of this presentation, I've described the way that we have investigated generating somatosensory percepts for bidirectional BMIs with an extremely multidisciplinary team. So we worked very closely with scientists, engineers, and clinicians. And that allowed us to work with a much broader population of people, as well as a broader set of electrode types. We were able to work with people with epilepsy, as well as with people with tetraplegia. And we worked with both ECOG electrodes that rested on top of the cortex, as well as intracortical microelectrode arrays, where we're targeting a much smaller area of cortex with smaller currents of electricity. And this, I believe this multidisciplinary approach allows us to better understand how cortical stimulation might be used to restore sensation in the case of a person with paralysis who has lost both the ability to move and to feel. So stimulation through of somatosensory cortex through ECOG and neuroport array clearly produces somatosensory percepts. And it's clear that we can target a specific somatotopy by choosing the correct electrode, and that we can generate qualitatively diverse types of percepts. And finally, it's interesting to note that there are different effects in terms of changing the parameters for these different devices and the different scales of current that we generate through them. For the ECOG devices, we found that larger parameter values yield more intensity, but can be less specific. Whereas for the microelectrode arrays, we found that proprioceptive percepts were more likely at higher amplitudes. And so in conclusion, all of these things help us to better understand how we might be able to generate these somatosensory percepts for bidirectional brain-machine interfaces. Thank you.
Hi, my name is Tyson Aflalo. I'm a research scientist and executive director of the Chen BMI Center at Caltech. I'm going to introduce you to a clinical trial in which we record simultaneously from posterior parietal cortex and motor cortex for neuroprosthetic applications. In way of background, the majority of neuroprosthetic clinical trials have focused on using neural signals recorded from the motor cortex. Motor cortex's descending projections are most directly responsible for cortical control of the details of movement during motor execution. However, motor behavior is not solely controlled by motor cortex, but is instead the product of an extended network that integrates information from our immediate visual environment, contextual factors, and high-level behavioral goals to determine which behaviors to execute. Over the past 6 years, we have conducted FDA-approved clinical trials that use neural activity recorded from arrays implanted outside the motor cortex to provide assistive technology. In particular, we have implanted the posterior parietal cortex, which I'll refer to as PPC. The PPC fits into this network as a critical node for transforming visual information into motor plans, as well as monitoring ongoing movements. In demonstrating neural control from PPC, our clinical trials have established that cortical regions of the motor network outside motor cortex enable closed-loop cortical control of assistive devices. Our results have shown that human PPC encodes many useful signals that include the trajectories of intended movements similar to motor cortex, but also many other action-related variables, including the goals of movement, hand shapes, cognitive motor strategy, action semantics, intended movements of multiple body parts, and even the decision of whether a visual stimulus has been seen previously. Neural representations of these diverse action variables are not randomly mixed at the level of the population, but instead are systematically organized around movement variables in an architecture we have termed partially mixed selectivity. Now that clinical trials have demonstrated neural prosthetic control signals from both PPC and motor cortex, an important question is how PPC compares to motor cortex. In what ways are the signals from the two regions similar, and in what ways might they be complementary, providing distinct types of information that can be used to enhance neural prosthetic function. We have recently initiated a clinical study in which we implant PPC and motor cortex in the same participants to allow a direct comparison. Here is participant JJ, our first subject with implants in both PPC and motor cortex. He sustained a spinal cord injury in March of 2017 and a go-kart accident. We use functional neuroimaging to help guide placement of Utah electrode arrays. The final implant locations correspond to the hand knob of motor cortex and the superior parietal lobule of the posterior parietal cortex. The implantation surgery was performed by Dr. Neuroprosthetics at UCLA. We have had good recordings from both brain regions, and we typically get a yield of single and multi-unit activity in the range of 100 to 150 units. Here is a visualization of sorted neurons for each array. Participant JJ started neuroprosthetic control sessions in January of 2019. I'm about to show you an example of closed-loop control from JJ's very first clinical session. This video was taken after about 4 minutes of total neuroprosthetic control experience, after some parameters were updated. Here we are combining activity from both brain regions. You can also hear JJ talk about what he is doing to control the cursor. Play video.
All right, so what are you thinking as you're moving the cursor around?
It almost feels natural. It feels like that I'm actually moving my wrist in order to guide the cursor. All the axes, diagonal, cross, back to center. As a matter of fact, the cursor itself is what's dragging.
After adjusting decoder parameters, training regimens, and patient strategies, within a couple of weeks, we were able to attain control performance that is remarkably similar to performance of motor intact individuals. Again, here we are combining activity from both regions. Play video. The prosthetic has worked great, allowing robust and versatile control across a range of experimental tasks and commercially available software. But for current purposes, the primary question is how the functional properties of the two brain regions compare. Our clinical trial has largely focused on using a variety of experimental paradigms to tease apart functional similarities and differences between motor cortex and PPC. For this webinar, I'll focus on experiments that have probed the timing of when intention-related activity is encoded. In motor intact individuals, one key feature distinguishing high-level motor intention regions like PPC from motor cortex is the existence of planning activity, or a neural coding of what an individual wants to do before they actually execute the action. Planning activity is generally demonstrated with delayed memory tasks such as we see here. In this task, a movement is instructed with a cue, the cue is extinguished during a delay period, and finally an instruction is given to initiate movement. In this case, with a luminance change of all targets. This task allows planning and execution to be dissociated in time. We know that motor cortex activity is largely expressed during motor execution. On the right, we are looking at some representative results from motor cortex. Single cell recordings in non-human primates and functional imaging in humans shows an emphasis on execution-related activity. In contrast, high-level regions like PPC, but also areas like premotor cortex, amongst others, encode the upcoming movement as soon as the participant is given information that informs the movement. The functional properties of motor cortex and PPC make sense given the location of these nodes in the overall motor production network. PPC takes in visual information and transforms this information into a motor plan, while M1 converts plans into coordinated movements. However, the functional properties that are identified in motor intact individuals may not generalize to patient populations that utilize these neuroprosthetic systems. This is because there are a number of critical differences between the two populations. For example, one key difference is the potential long-term plasticity that might result from spinal cord injury. Another key difference is that the neuroprosthetic can fundamentally alter how a cortical region contributes to motor behavior. Let's take the case of PPC. PPC is a region that traditionally plays a role in preparing movements. However, in a neuroprosthetic system, PPC is asked to directly produce movements of a neuroprosthetic effector. In this way, we fundamentally change the causal contribution of PPC to the production of motor behavior. This, of course, will depend on how exactly neuroactivity is read out from these brain regions. Through the course of the experiments we are presenting in JJ, we have used both motor cortex and PPC as motor output regions. In other words, our algorithm directly translates moment-to-moment fluctuations in neural activity into force-like signals that update the instantaneous position of the cursor. Thus, due to the way we interpret PP signals, we truly are modifying its causal contribution to behavior. To assess the basic timing of signals in motor cortex and PPC, we had JJ perform a delayed memory task using the neuroprosthetic system. Thus, you would guide a cursor to the cued target under brain control after the delay. Here I am showing the results from the very first session we ran a delayed memory paradigm. We are showing the percent of the population that demonstrates significant coding of movement direction as a function of time in a sliding window analysis. The window is around 300 milliseconds. This is an offline analysis applied to the data that was acquired during closed-loop cortical control. We see that PPC encodes movement immediately after the cue, instructing movement direction, and this coding continues through the time of movement execution. In contrast, motor cortex is largely restricted to the time of movement execution. Thus, despite the spinal cord injury, at the start of our clinical trial, responses are exactly as predicted based on motor intact individuals. We then repeated the delayed memory task at sporadic intervals over a 10-month period. Over this time, the subject attended sessions regularly, about twice a week, and performed a wide variety of neuroprosthetic tasks that typically involved continuous interaction with a prosthetic interface. These tasks typically did not involve enforced delay periods, but instead continuous control. The question was, would long-term use of a neuroprosthetic change the core functional properties of the timing of intention-related signals? So here we see the results of the same task performed about 10 months later. The functional properties of both regions are preserved. This result, amongst other early results, made clear that functional distinctions between motor cortex and PPC are preserved in the clinical population and provide distinct and complementary representations of motor variables. The fact that they preserve their properties suggests that network connectivity provides a strong constraint on the types of activity patterns that these brain areas can express. This is not to say that the functional properties are completely immutable. We have found that a contextual manipulation can modify the degree to which motor cortex expresses activity during the planning period of the task. I'm going to present the results of the delayed memory paradigm with this contextual manipulation. On the left, I show what we call a brain-coupled version of the task, the results for which we were shown above. In the coupled version, the neuroprosthetic system is always on. That is, neural fluctuations are directly translated into cursor motion throughout the entire interval of the trial. On the right, I show a schematic of what I call the brain decoupled version of the task. In this version, the neuroprosthetic system is essentially turned off except for the go phase. That is, neural fluctuations are decoupled from cursor movement during the cue and delay periods. What we find is that when brain activity is coupled to cursor movements, we see very little motor cortex activity during the cue and delay. In contrast, when the brain is decoupled from cursor movement, M1 develops activity. These changes can be observed through the course of a single session. We think this is likely the natural consequence of certain optimality principles at play, though I cannot fully elaborate the result due to time constraints. However, I think it's important to show that motor cortex activity can express planning-like responses, but only in certain limited and unnatural contexts. Finally, I'll highlight two complementary properties of these brain regions for closed-loop continuous control. One motivation for implanting a high-level region like PPC is that it may enable faster decodes. From the delayed memory task, we see that PPC activity encodes the participant's intentions much earlier than motor cortex. However, the enforced delay period exaggerates the differences in the timing of PPC and motor cortex activity. Due timing differences persevere when the subject simply sees a stimulus and reacts to it. We are interested in intent, but not, for instance, visual transients. To ensure neural population activity is encoding generalizable intention signals, we had the subject perform neuroprosthetic movements in a task at interleaved pro-trials in which a change of color at the peripheral target was used to instruct movement, and symbolic trials in which an arrow at the center location instructed the target. This allows us to cue the same intention with distinct visual cues. The subject performed this task in closed-loop brain control. In offline analysis, we used a sliding window cross-condition classification analysis. That is, we trained the classifier on the symbolically cued trials to predict the provert trials and vice versa. This procedure ensures that the classifier cannot cheat and use visual information to boost accuracy. For simplicity, here we are looking at the averaged across condition accuracy, and we see that both areas generalized pretty nicely, but that PPC encodes the intentions about 100 milliseconds faster than motor cortex. So from this perspective, PPC is better than motor cortex. Conversely, we find that M1 better tracks the intentions of the subject for long sustained movements. To demonstrate this point, the subjects performed a simple cursor control task, but we slowed the cursor such that movements took on average about 2.5 seconds to complete. Here we are looking at cross-validated classification accuracy as a function of time using a sliding window analysis. We see that M1 sustains its selectivity through the full duration of the movement, whereas PPC activity attenuates. Thus, motor cortex better represents the moment-to-moment intention of the participant through the full course of these slowed down movements. From this perspective, motor cortex is better than PPC. I often get asked the question, how does the brain area that you record from, or does the brain area you record from really matter? The human brain is the most powerful statistical learning machine we know of, after all. But we find that the core functional properties of brain regions are largely preserved, despite injury and novel functional demands. And further, that brain regions have distinctive and complementary properties. Now, there are a lot of different ways that a neuroprosthetic system can be built, and it is clear that brain areas should be selected and decoding algorithms tailored to harmonize with these different implementations. And I also think that these findings truly stress how neuroprosthetics is a multidisciplinary field. It requires an understanding and study of the brain to understand what properties are available for decoding, combined with an ability to decode these properties in real time from populations of neurons, and a means of translating these decoded signals into meaningful assistance. These require the disciplines of systems neuroscience and cognitive neuroscience, statistics and machine learning, as well as engineering.
Hello, I'm Charles Liu. I'm a neurosurgeon with an interest in neurorestoration. My role is to manage the clinical aspects of this complex interdisciplinary project. So in this talk, I'll briefly discuss some of the clinical facets of this complex interdisciplinary project involving long-term surgical implants in human participants, briefly touch on the clinical needs for transformative solutions to spinal cord injury. We'll talk about the complex clinical resources that are necessary for efficient interdisciplinary research. I'll make some comments about how we go about identifying and recruiting the ideal research subject, and we really consider these subjects research partners of ours. There's not a lot of information about how to do the surgeries properly, so I'll make some comments about the surgical considerations for the optimal implantation of these microelectrode arrays. And then I'll also touch on some of the ongoing research subject needs until the explantation is done and the subjects exit from the project, as well as what their needs are beyond that. So it's important to consider the public health dimensions of spinal cord injury. Here are the latest statistics from the National Spinal Cord Injury Statistical Center at UAB. I've circled some of the important points on the slide. You can see that there are almost 20,000 new cases of spinal cord injury per year. And in the United States, there are approximately 300,000 people living with spinal cord injury. So we're talking about something that's certainly not an orphan disease. Again, this is a busy slide, but more statistics on spinal cord injury. I want to focus the audience's attention on the numbers circled in red. This relates to the costs, both in monetary terms as well as in human terms. You can see that the lifetime cost associated with a person who becomes tetraplegic at age 25 is somewhere between $4 to $5 million, depending on their level of injury. And you can also see with the numbers circled in the bottom red circle that these patients, these people can live for many years, which leads to considerable human suffering costs. It really, as a result of this, that the potential for transformative solutions, whether they're found in regenerative medicine or in neuroprosthetics research, such as this current project, and this is very much in the public consciousness. And that as a clinical person working with spinal cord injury people, I get asked about these things all the time, and you see that it's actually made its way into the magazine-type publications. want to spend a few minutes and just go over what the resources are necessary, the clinical resources are for accomplishing this type of research. Complex studies such as ours require acute care resources, and most of these can be naturally found in centers where there are active clinical BCI programs, and these are done for a variety of indications such as movement disorders, epilepsy, pain disorders. For the current project, these resources are found at USC Keck Medical Center, and in the slide you see two schematics, and one of course is of a deep brain stimulator where penetrating electrodes are placed deep into the brain and attached to pulse generators for indications such as movement disorder. And you see the image in the bottom right corner of NeuroPace, which is a bidirectional device that records the seizure activities from implanted electrodes and applies the stimulation to ablate those seizures. Beyond the resources required for regulatory oversight, such as IRBs, as well as those required for the perioperative resources for implantation, there are additional resources, such as those required for recruitment of study participants and resources to manage their ongoing needs. We have embedded our study at Rancho Los Amigos National Rehabilitation Center, which here is shown in a beautiful image here that this center was newly rebuilt recently for close to $500 million. Rancho is one of only 14 National Institute of Disability, Independent Living, and Rehabilitation Research funded spinal cord injury model systems. And it's the only one that's found in the West Coast. You see here really Rancho in the green, the red arrow pointing to this green dot here is the location of where our rehabilitation center is. Here is a view of the major public space in the new outpatient building of Rancho. This is the space that sees the most traffic from all types of, all members of the Rancho Los Amigos community. When the building was created, an artist, Deb Ashram, was commissioned to create art for the space, and she chose to put Rancho's history on the wall. And you can see that our project is indelibly represented here. You see this red arrow pointing to this image, and Rancho really has a wonderful history, and the current project is now a very has been recorded as being a significant part of it. And of course, here is just a close-up on the image that was pointed at in the last slide. The artists also created a wall with video screens playing clips of ongoing projects, and our work continues to be represented. This is significant in that the Rancho community can see the research that's going on to solve problems that's very personal to them. And in that way, they can potentially be interested in participating in these projects for recruitment purposes. We have a very robust outreach program. Here in this slide, you see Dr. Anderson moderating a discussion between two study subjects with members of the community of Rancho Los Amigos, this community of peoples with disability. And you can see in the room there are many people there who are tetraplegics. This new patient expo will be held annually, and this makes the research programs reachable. And in this way, we've embedded our research into the community, into a veritable hub of resources for patients with disability. I'm going to next touch on some of the issues related to the surgical implantations. There is not much collective experience with the implantation of UTAR arrays in humans. And much of the science that we're talking about here depends on the quality of the implants. We've certainly learned a lot in just the few implant surgeries in humans that we've done. So I think it's worth making some comments. This is what the typical OR for an implantation looks like. I'm here on the left with the black cap. The person in the middle is Dr. Brian Lee. The person on the right is Dan Kramer, Dr. Kramer, and both Brian and Darren, and pardon me, Brian and Dan are alumni of the Anderson lab. There are quite a lot of differences in the implant surgery in humans that I think is worth commenting on. The first significant difference that we've noticed is that in humans, the leptomeninges can be quite thick, and you can find this thickening especially near the veins. This makes it potentially very difficult to insert the arrays. We have now modified our implantation technique by removing the arachnoid microsurgically, and what you see in this next video clip is just how thick the arachnoid can be and what the technique to remove it looks like. Go ahead and play the video. So the other major issue that we identified with the implantation of the microarrays is that very often the location of the ideal implant area can be obstructed by relatively large cortical arteries and veins. But this can be readily managed safely by a skillful neurosurgeon with good command of microsurgical techniques. And in this next video clip, you'll see us actually moving a good sized artery because it's in the way of our implant. Go ahead and play the video. So finally, when the areas of implant are exposed, the arrays are inserted into the cortex using a pneumatic inserter that's held by a stereotactic frame. Here you see what this looks like. You notice in the downward red arrow pointing at the brain shift that makes the management of the very stiff gold wire leads even more challenging in humans. So I wanted to just show a quick video clip of what the pneumatic inserter implantation process looks like. In this next video clip, you'll see that we brought the pneumatic inserter directly over the microelectrode array, and you're going to see that very quickly it's going to essentially insert the microwires into the cortex. And we're going to play the video twice because it's really easy to miss it. So this slide shows that the surgical plan that has been identified by the research team, that created by the research team, with the position of the microelectrode arrays has been perfectly executed. You notice that the S1 arrays are implanted right next to the large vein where the thickened arachnoid was removed in the first video clip. And the PMV array is implanted next to the loop of artery that was moved, that was shown in the second video clip. It turns out in this particular implant, all of the arrays are near vessels, and certainly that was not our choice, but it just so happened that these are the places that we wanted to put the electrodes. Want to just briefly touch on the ongoing study participant needs. After the perioperative period, we found that progress of our research can be limited by both participants' physical and social well-being. And this is certainly not trivial in this population. These are very fragile patients. We're very delighted to find that all of our patients have enjoyed the project, and we continue to find ways for involvement even beyond explantation, and the study is concluded for the participants. So to summarize, efficient neuroprosthetics research requires optimal management of complex clinical resources. This can be achieved by embedding advanced research into the ongoing activities of a central hub for people with disability, and we've done that with Rancho Los Amigos National Rehabilitation Center. The neurosurgical aspects of neuroprosthetics implantation requires further refinement, and this refinement can lead to further efficiency in terms of advancing the scientific goals. Finally, I just want to acknowledge the institutions that are involved with the USC Neurorestoration Center. The clinical resources described here in this talk are drawn across a large number of collaborators of the USC Neurorestoration Center. Thank you.